Wonders of Medicine, Part 1, Basics of Drug Therapy, Unit 1, Receptors as Drug Target. How do drugs actually work? Scientists have long tried to answer this question. They eventually realized that in order for a drug to exert its selective action, human cells must have a site for the recognition and acceptance of a specific type of drug. This site of the drug action is termed a receptor. A drug-receptor interactions have now become the focus of research for the development of new drugs. It was Paul Ehrlich, a German chemist, who first proposed the concept of receptors. His observation of drugs used to treat parasitic infections led him to conclude that each cell would have characteristics that allow it to recognize particular molecules. He proposed the lock and key hypothesis that a drug binds to a receptor in the same way that a key fits into a lock. This was based on his belief that the drug and the receptor must be structurally complementary in order to recognize each other and initiate an effect. Receptors, which are the primary targets of the majority of drugs and are biological macromolecules that have evolved specifically for intercellular communication. This type of receptor includes ion channels and proteins in the plasma membrane. Basically, the biological function of this receptor is to respond to the body's own endogenous chemical messengers, such as hormones or neurotransmitters. When binding takes place, it triggers a series of biochemical and physiological changes known as a response. Other types of receptors are the macromolecules, which are essential to a cell's normal function or replication, such as enzymes, lipids, and nucleic acids, particularly DNA. Drugs which bind with receptors and mimic the effect of hormones or neurotransmitters are called agonists. Drugs which bind to the receptors but do not have the unique structural features necessary to activate them are called antagonists. Science thinks the antagonists occupy a binding site of the receptors. They prevent activation of agonists. Advances in the sequencing of the human genome have enabled researchers to identify the structure and varieties of receptors. Many newly discovered receptors have become potential targets for further pharmaceutical development. G-protein coupled receptor, GPCRs, and ligand-gated ion channels are two of the most important receptor types now being targeted in the quest for new clinical use for drugs.